Okay, so this PowerPoint presentation is for Chapter 10, which is on cell reproduction. We're going to be covering a specific type of cellular reproduction here called mitosis. And we're only covering part of this chapter, the first two sections. That's going to be what's important for both lecture as well as lab. So let's go ahead and get started. So this image is right from your textbook. I really like how this image shows beautifully some cell division. The first two images are, all three images are the sea urchin. Okay, so this is an animal that lives in aquatic environments. It's multicellular. And if we take a look at the first two images, those are taken with an electron microscope. So very detailed, high resolution, nice images. This specific type of electron microscope would be the scanning or SEM. Remember we learned about that a few chapters ago. And then the last image over on the right is just a regular photograph. Okay. So again, this is the sea urchin and it's showing mitosis. In this first picture here, the first image, we have what was a diploid zygote or a fertilized egg. And we're going to learn what diploid means, but just to give you a kind of a heads up. Well, first of all, a zygote. So the zygote is the result of fertilization. So it's the result of sexual reproduction. So you have a sperm from the male parent that is going to fertilize or combine with the egg from the female parent. Now, each of those structures, they're called gametes, by the way. Each of those structures is going to contain half of the genetic information that is present in the organism's somatic cells or regular old body cells. Okay, so you have these egg and sperm, which again are called gametes. They're going to contain half the genetic information that's normally present in that organism. And so they're going to be called haploid cells. Okay, half the genetic information. Those two cells come together. That union is called fertilization. And that first cell, the result of that fertilization, is called the zygote. Now the zygote is going to have chromosomes now from each of those parent cells. And so they will go back to the normal chromosome number. So the zygote is going to be diploid. Or it's going to contain two copies of each chromosome. One that was inherited from the egg, mom, and one set that was inherited from dad, right, the sperm cell. Okay, so we end up with a diploid zygote having two copies of each chromosome. That zygote is then going to undergo cell division, which again is called mitosis. And so in this first figure here, this is showing that first round of mitosis. So we have one cell, which is splitting now into two. Okay, during early developmental or embryonic stages here, those cells are going to actually stay together. Later on, sometimes cells will actually kind of break apart from each other, or move apart from each other, I should say. These will eventually become two separate cells, but they may remain attached to each other. So both of these cells now are going to divide more. So mitosis is going to happen. It's going to happen again and again and again. And what we show here in this second picture, the second electron micrograph, is something called the 16-cell stage. Okay, so multiple rounds of mitosis has led to this. If we count up all the cells in this ball of cells that we have here, there would be 16 cells. Okay, and then mitosis continues, and we get what we call cellular differentiation. Cells start to take on, jobs or roles. They become specialized. And eventually, you end up with a complex, multicellular sea urchin that has specialized cells and tissues, and maybe even there's going to be even some organ systems in this organism. Okay, pretty advanced and complex. Again, how does an organism go from a single-celled zygote to a multicellular organism? Mitosis. So that's what we're talking about here. It's all about cell division. Cell division is going to be required for growth. Okay, and if we come down here to my second bullet point, this cell division that we're talking about here, we're talking about early development, but you need to know that mitosis is a continual process. Mitosis will continue throughout life. Right inside of you right now, some of your cells are going through cell division. They're going through mitosis. Why? growth, right? Repair. If you have tissue damage, mitosis is going to occur to replace those cells that have become damaged and have become lost. Maintenance. Okay, there are some tissues in your body that are continually being renewed. The best example is your skin, right? You know that you lose those top layers of skin. You've probably seen it. If you just kind of take your arm and rub it right now, you can see kind of some white flaky material coming off, especially if your skin's dry, which mine is right now. What's happening there is you're losing that upper layer of cells. Those cells need to be replaced. And so mitosis is going to occur. That's going to replace those cells. An important thing to remember, and we're not going to touch on this part of the chapter, but I welcome you to read it if you have time. Cell division or mitosis must be closely, tightly monitored or regulated. And so your cells have mechanisms in place to really monitor this process to make sure that mitosis is controlled. When a cell's cell cycle loses that regulation, it can be very, very dangerous. Okay, we've all heard of cancer. Cancer is the result of loss of cell cycle control. Okay, you have cells that are not supposed to be replicating and dividing that are, for whatever reason, replicating and dividing, usually pretty rapidly. Okay, and that's really dangerous. And the last bullet point here, again, the end of the chapter touches on this. We aren't going to have time to do that. We've got to fit both mitosis and meiosis in this week. So again, I invite you to read that part of the chapter, but I won't be testing you on that. So single-celled organisms, we've learned about these by now, right? So we've heard of prokaryotes, right? Organisms that are single-celled and that lack membrane-bound organelles, including a membrane-bound nucleus. Okay, those are what prokaryotes are. They're going to reproduce asexually, pretty much by mitosis. We give it a special name. It's called binary fission. Okay, we are going to look at prokaryote genetic information just to kind of touch on it. So if you do read that last part of the chapter, it should make sense to you. But again, we don't have a lot of time, enough time to get to that. Okay, so again, reproduction and single-celled organisms like bacteria, right, that are prokaryotes, they're going to reproduce basically by mitosis, which gets a special name called binary fission. Okay, so the first part of this chapter, the first section of 2 that we're going to cover, is titled Cell Division. So this is 10.1. Here's a list of learning outcomes. These are what we expect to get out of this part of the chapter. We're going to take a look at both prokaryotic and eukaryotic genomes, and hopefully you've defined that term genome by now, but I'll go ahead and I'll define it for you as well, just to reinforce that, right? I really believe in repetition, as you can see, right? We do the same set of assignments each week, and it seems repetitious, and it is for a reason. Repetition helps us to remember. So that's the first thing we're going to do. Then we're going to distinguish between chromosomes, genes, alleles, and traits. And we're going to talk about how chromosomes become compact and why. Okay, so let's start off by first defining the cell cycle. So what exactly is this? The cell cycle is going to be a sequence of events or stages, and we're going to look at these stages in the next part of the chapter, 10.2. So it's a sequence of stages that take place in a cell's life as they go from a parent cell to 2 genetically identical daughter cells. Okay, so the cell cycle, we're going to go from one cell, which we're going to call the parent, and that cell is going to divide into 2 daughter cells. I'm not sure why we call them daughters and not sons, but we do. Okay, those 2 daughter cells are going to be identical to each other. They're going to have exactly the same genetic information, the same number and types of chromosomes. Okay, it's pretty much like cloning, right? You go from one parent to 2 cells in the end that are going to be identical to each other. They are clones of each other. Okay, so again, we're going to dive into the cell cycle in the next part. We're going to talk about mitosis. We're going to talk about what comes before mitosis, and we'll talk about what comes after mitosis. Mitosis is going to be a set of stages made up of prophase, metaphase, anaphase, and telophase. Okay, and that set of stages is going to divide the nucleus, and then that's going to be followed by something called cytokinesis, in which the rest of the cell is divided. The organelle, the cytoplasm, that plasma membrane gets divided. We end up with 2 cells in the end. But again, before we take a deep dive, we have to look at genetic material first, and this is pretty interesting. Okay, so first off, I didn't skip anything. Okay, so first off, what is genetic material? We're talking about DNA, right? Deoxyribonucleic acid. You were introduced to this a couple of chapters ago, so here we have to kind of refresh our memory. DNA is going to be the genetic material found in a cell, or we could say the cell's genome. Okay, all of the genetic material found in a cell is the organism's genome. DNA is always going to be double-stranded. Okay, it's a double-stranded molecule. It's made up of a sugar phosphate backbone with nitrogenous bases on the inside. Those bases are adenine, thymine, cytosine, and guanine. Maybe you've heard of those. Okay, it's an amazing molecule, and if you take General Biology 2 with me, we'll get really deep into what that molecule looks like, properties of it, how it replicates, what the cell does with it. Okay, so that genetic material in prokaryotes is going to be found in a region of the cell called the nucleoid, but not in an enclosed membrane. So remember, prokaryotes lack a nucleus. Okay, so where is their genetic information? Well, it's in the cytoplasm, but it is found concentrated in a region called the nucleoid. Okay. In prokaryotes, this is really neat and important. In prokaryotes like bacteria, they're only going to have 1 chromosome, which is pretty neat, right? That 1 chromosome is going to be double-stranded, and it is going to be circular. So we have a circle of genetic information in bacteria. And you can see that here. This brown material, this is the DNA, and it's just one, it looks kind of bunched up, but it's just one circular piece of DNA. Okay? 1 chromosome, double-stranded, circular. Again, where is it found? It's found in the nucleoid. It's a region in the cell, but there is no nucleus. Okay, now here's just one other thing I want to allude to. Again, if you go on and you take General Bio 2 with me, we'll talk more about this. Now, although bacteria and prokaryotes in general only have 1 larger, circular, double-stranded chromosome, they may have these small, little circular, double-stranded bits of DNA called plasmids. Okay? And those plasmids are not required for the bacteria in general. However, the plasmid may carry some pretty cool genes on it that might help the bacteria be able to survive or grow or really excel at living in particular environments. So for example, you've probably all heard of antibiotic resistance in bacteria, right? You've all heard of that. A common one is MRSA, methicillin resistant Staph aureus. That's a really common one that's unfortunately pretty rampant in many of our hospital settings. That particular antibiotic resistance is caused from a gene that's found on one of these plasmids. And, unfortunately, these plasmids can be passed between bacteria. So they can actually share those little circular pieces of DNA. So that antibiotic resistance can spread from one bacteria to another relatively rapidly. Okay? If you take General Biology 2 with me, we're going to actually work with plasmids, where we take bacteria, right? We work with E. coli, and these E. coli, they cannot grow in the presence of antibiotic, and if I were to shine an ultraviolet light on them, they're not capable of glowing. However, we're going to take a plasmid, a little small circular piece of DNA, that has 2 really important genes on it, the antibiotic resistance gene, as well as, it's called green fluorescent protein. I like to call it the glowing gene. We're going to take that plasmid and we're going to insert it into our E. coli. And we're going to grow that E. coli under very specific environmental conditions. And some of our E. coli will pick up that plasmid and will express those genes. So they'll be able to grow in the presence of antibiotic, and they'll be able to glow in the presence of ultraviolet light. It's pretty easy to get bacteria to take up plasmids. So it's kind of scary. Okay, so again, DNA is genetic material. We can also call that an organism's genome. It's double-stranded, and in prokaryotes, which usually have just one single chromosome, that chromosome is going to be circular. Now that's very different, let me move my camera here, in organisms like us, or plants, or fungi, or even protists, okay? Any organism that is eukaryotic would fall into that category. And eukaryotes, yes, they're still going to have double-stranded DNA, but that DNA is not circular. Instead, it's linear. And most organisms, most eukaryotes, are going to have several of these linear chromosomes. The number of chromosomes depends on the species. We'll use humans, right? Because that's a great example. Humans are going to have, in all of our cells that are not involved in sexual reproduction, so we're excluding our gametes. So egg cells for females and sperm cells for males. So let's not talk about those for a minute. In the rest of your body cells, you are going to find, if you were to pop open that nucleus and take a look, you would find 46 chromosomes. So we say that all of your somatic cells, all of your body cells, not including your gametes, are diploid. They contain 46 chromosomes, or we could say 2 pairs. Okay? We can use the term 2N to represent diploid, or having 2 copies of each gene. Where did those copies come from, right? Remember that you started off as one single-celled zygote. You got half your chromosomes, or one set, from your mom, and half of your chromosomes, or one set, from your dad. Now they end up in this cell together. You have a total of 46 chromosomes. 23 of them came from your mom, and 23 of them came from your dad. And now they are chromosome pairs, okay? We'll get to pairs in just a second. So let's make sure we got that. So any eukaryotic organism, we're going to have several double-stranded linear chromosomes. That number depends on the species. In humans, that's going to be 46 chromosomes for our somatic cells, and our gametes, our sex cells, egg and sperm cells, they're going to have half that number. So we say that our gametes are haploid, or 1N, and that number for humans is going to be 23 chromosomes. Okay? So remember again, upon fertilization, so we have egg, 23 chromosomes, sperm, 23 chromosomes, they come together. We go back, we restore that number to 46. So 46 or 23 pairs of chromosomes. Those pairs of chromosomes get a really special name. Look here, this term homologous. So homologous pairs of chromosomes. What does that term mean, homologous? It means that those chromosomes, right, so let's compare, let's say chromosome 1 that came from your mom and chromosome 1 that came from your dad, they're a pair. Okay, they're not going to come together. They're going to stay separate, but they are a pair. What does that mean? They're homologous to each other, which means that they're going to be the same size, the same length, and if I take a close look, they're going to carry the same genes. Okay, which means they're going to have information coding for something at exactly the same location or locus, position on the chromosome. Now it's important to keep in mind here, just because they carry the same genes doesn't mean that they code for the same, or doesn't mean that they have the same allele. What does that mean? So let's go back. Let's say we have chromosome 1 from mom and chromosome 1 for dad. And I look at a specific genetic locus and I find the gene that codes for, let's just say eye color. Okay? I find the gene that codes for eye color. So that's the gene in that specific location. That's the trait I'm looking at, eye color. Well, let's say your mom gives you the allele, the form of the gene that codes for blue eyes. But your dad gives you the allele or form of the gene that codes for brown eyes. So we have the same genes, it's the eye color gene, but we have different forms or variants of that gene. We call those forms or variants, We call those alleles. So what color eyes would you have? So mom gave you the allele for blue. Dad gives you the allele for brown. What color eyes would you have? Hopefully you answered brown. So for eye color, brown is going to be dominant to blue. Okay, so you would have brown eyes. However, you could have children that have blue eyes because you carry the gene, the form of the gene, the allele, that codes for blue. Okay, very interesting, right? Genetics. Again, in general biology 2, we spend a couple of weeks in lab and in lecture talking about genetics, talking about Mendel and how genes interact, and talking about dominance and recessive. Okay, so here I'm just kind of introducing it to you, but again, we would get a little deeper into that in general bio 2. Up here, this picture here, this is really neat. You're going to see this, or you have seen it, and you will see it again in lab. This is a micrograph, so an image taken with a microscope of a plant. This looks like the root, where we have some mitosis occurring. Okay, we haven't talked about the steps of mitosis yet, but I want to point out some cells. So a lot of these cells, if I look here, this is one cell. You can see this square to rectangular shaped border. That's the cell wall. And you can see the circle in the middle. That's the nucleus. And you can see it looks kind of grainy, and we've got these dark spots. This cell is probably not dividing right now. Instead, it's pre-division. So we would call this cell an interphase cell. Well, look here. This cell looks really different, right? So we've got, again, our cell wall, that outer border, and it looks like the nuclear envelope or nuclear membrane has kind of faded away, and I'm left with this stuff inside that looks like it's thickening. That's the genetic material starting to condense into chromosomes. And we'll talk about how that happens in just a minute. But this cell right here would be in prophase. This cell down here, look at this. So we've got our cell wall. Nucleus is not present. Chromosomes are very thick, and they look like they're lined up in the middle. That's called a metaphase cell. And the same thing, this cell down here, these chromosomes look lined up in the middle. That's a metaphase cell. Take a look at this cell right here. This looks like it just divided into two. Looks like this is the tail end of mitosis. So we might say that this is cytokinesis, and we're about to produce two totally separate cells here. Okay, pretty neat. Okay, so I think we covered everything on this slide. I actually want to pull up this just in case. Okay, so let me move my picture up here. Okay, so this is a neat image here. This is called a karyotype. So if we take a look here, this is the nucleus of the cell, and we're kind of pulling out the chromosomes. And if we were to count these up, which we have, we've actually not just counted them up, but we've put them homologous pairs next to each other, which is pretty neat. We can actually see the 46 total chromosomes, or 23 pairs of chromosomes. Now this cell is going to be kind of paused after chromosomes have condensed, and now we're kind of taking a look at that cell. And this particular cell here is a female somatic cell. Okay, so again, that's why there's 46 chromosomes, not just 23, right? And it's a female somatic cell, and we'll talk about why in just a second. So let's take a look here. We've got pairs 1, pair 2, 3, 4, all the way up to 22. Okay, and if we look at the 23rd pair, right, chromosomes 45 and 46 here, you can see that they're the same size and length and all that. These are going to be X chromosomes. And so when we say that this is a female somatic cell, females inherit 2 X chromosomes, where males inherit an X and a Y chromosome, which is kind of weird. So if this were a male these two chromosomes here would actually be different sizes because the X and the Y are different sizes. The Y is a little bit shorter. Okay. Now, if you take a look at these homologous pairs, notice each pair, right, if you look at the two chromosomes of the pair, they're the same size, the same length, okay? They've been dyed the same color. Chromosomes aren't actually colorful, right, under the microscope unless you provide some type of a stain. Okay, again, this is called a karyotype. It's where we can actually physically look under the microscope at your chromosomes. Pretty neat. Okay. I wanted to give you just a little bit more information about these chromosomes. So again, I alluded to this, but I wanted to make sure I put it down in words as well. So during sexual reproduction, right, egg and sperm comes together. So you've got a cell containing 23, or for humans, 23 chromosomes. We call that the maternal set of chromosomes. And then you have a sperm cell, right, carrying the paternal set of chromosomes. They end up in one cell, that's called the zygote, and they end up as pairs. Okay, let's take a look again at chromosome number 1, let's say. Chromosome pair number 1, one of these came from your mom, or the mom of the cell, and one of these came from the father of this cell, right? So we have the maternal chromosome and the paternal chromosome, and together, collectively, they make chromosome pair number 1, a homologous pair of chromosome. Again, they're the same size and the same shape, and they would have the same genes at the same locations or loci. However, remember again that they may have different forms of the gene. So we just talked about eye color, right? If mom gives the allele for blue eyes and dad gives the allele for brown eyes, those are two different alleles of the same gene. We would have to figure out which one gets expressed, which one doesn't, or do they both? It depends on the gene. In that case, the brown would be expressed and the blue would be hidden. The same thing is true for other human traits that we could spend hours talking about. Again, if you take GB2 with me, we will spend hours talking about them. But if you think about blood type, right? Go ahead and ask your parents what their blood type is. And depending on what their blood types are, you may have the same blood type or it may be different. So for example, if your mom is AB and your dad is A, you could be type B. Okay? Or you could be type A. You cannot be type AB. And there's no way that you could be type O. Okay? Again, that's genetics, which we talk about in more detail in Bio 2. Same thing with other traits like widow's peak, or whether or not you have mid-digit hair, or whether or not you can roll your tongue, right? All of that are examples of genetic traits in which you can inherit different alleles, and that will lead to genetic variation, right? You may not have the same blood type as either of your parents. Okay, how is that possible? It is, right? You may not have the same blood type as any of your siblings. You may not, you may have blue eyes and one of your siblings may have brown eyes, and maybe both your parents have brown eyes. That's also possible. Okay? All right. So it leads to genetic variation. Sexual reproduction leads to genetic variation. Okay, so let's get into chromosome packing. So if I were to take one of your cells and open up the nucleus and take your genome, all the genetic material in that nucleus, and stretch it and spread it out, it would be about, in length, about 2 meters or 6.5 feet in length. That's a lot of genetic material, right? Your cells are only 10 micrometers in diameter. How do we get all of this, 6 feet of genetic information packaged up into a little tiny cell inside the nucleus? Again, your DNA is not stretched out inside of your nucleus. It's not, end to end. Instead, your chromosomes are going to be tightly packaged. Okay? Your genetic material is never just willy-nilly spread about. It's always packaged to some extent, sometimes more loosely, and sometimes very tightly, depending on which part of the cell cycle we're in. into these structures called chromosomes. Okay, and there's going to be really important proteins that play a really significant role in that packaging. So let's talk about the levels. And I'm going to just move my camera so that you can still see me, but put me off over to the side. So the first level, one second here. It's giving me a hard time moving my camera. I'm not sure if it's actually moving or not. So hopefully you can see everything on the screen. So the first level of chromosome condensation, or compaction, not condensation, compaction or packaging, is going to involve really specific proteins called histone proteins. And if you take an advanced level of biology, you'll learn about the number and types of histone proteins. We're just going to kind of keep it simple here. So if we take a look here at the first level here, this is just the DNA double helix, okay, not packaged. We will take the DNA double helix and we're going to wrap it around this central core here, which is the histone protein. Now the different colors again represent the different types of histone proteins. We're not concerned about the different types. In general, these all belong to that class of proteins called histones. So that first level of chromosome compaction or packaging is taking that linear double-stranded piece of DNA and wrapping it around a core of proteins called histone proteins. And if you look down here at this next one, the next level down here, what we have here is what we call the beads on the string. So you have a nucleosome, which is DNA wrapped around histone proteins, and then a little tiny space, and then the next nucleosome. Again, DNA wrapped around histone proteins, and then the next little space. That space is called linker DNA. Okay, so you have nucleosome, linker, nucleosome, linker, nucleosome, linker. It looks like beads on a string, right? Those beads are called nucleosomes. What are they made up of? It's just a core histone protein with DNA wrapped around it. Then there's a little space, and then another core histone protein with DNA wrapped around it. Okay, so again, your DNA is never just kind of naked, willy-nilly, just spread out. Instead, it's always coiled around these histone proteins. Now, when DNA is replicating or a gene has to be transcribed, if a protein has to be made from your genetic material, those histone proteins can move, they can slide, they can come unwound to give better access to that DNA. Okay, but in general, your DNA is going to be tightly associated with these proteins. This tight association with these histone proteins gives DNA the next level of organization, which we called chromatin. Okay, so if you hear that term chromatin, which you will in here and in lab, chromatin is DNA associated with proteins. And we know now that those specific proteins are called histone proteins. Okay, now when your cell goes to divide, it has to compact and organize that DNA even further. So this second level, okay, is the beginning of forming that tightly coiled and compact chromosome. We have further condensation. It's coiling into what we call the 30 nanometer chromatin fiber, and that's what's shown here down in box 1, 2, 3, 4 of this figure. So we take those beads on a string, right, that chromatin, and now we start to loop it and condense it even further around these proteins called, I don't know if the name is on here, they're called condensin proteins. Okay. Now those get even more tightly coiled and even more proteins become associated until we end up at our third level of chromosome compaction, where we have actual chromosomes that are completely done. And what we're seeing down here, this last box, this particular chromosome here is formed after DNA replication. And you can see it has one complete arm there, and then two. Here's that second complete arm here. This is called a dyad chromosome or two-armed chromosome. And in the center here, these yellow things, where there is this cinching or this bringing together of this chromosome, that's called the centromere. Okay, again, you're going to hear some more of these terms in a minute, and you'll see them in lab as well. The arms here are exact copies of each other, and they're called sister chromatids. Okay, hopefully that's a term that you read about in the chapter. So this is how our chromosomes become compact. We go from a double-stranded linear piece of DNA to nucleosomes, or beads on a string. Okay, that's that first level. The second level, we start to coil that up even more into the 30 nanometer chromatin fiber, which then becomes even more compact, and that last level is actually called the chromosome. Okay, again, why does our DNA need to do this? To stay organized, especially during mitosis and meiosis, so especially during cell division. But even, when the cell is not divided, if your chromosomes, or if your, genetic material, your DNA, is just kind of spread out all over the place, there's a higher chance of damage, breakage, to that DNA, which can be very, very dangerous. Okay, so your cells are always going to organize that genetic material. Okay, so again, I hope I'm trying to move my camera around so that you can read everything on these slides. So at this point, I want you to pause, again, the video. I want you to take out your Are You With Me sheet, and you've got terms to define first, followed by a couple of questions. Now again, these questions have sketches required, just like you've been doing the past couple of weeks here in lecture. Make sure that you are actually sketching as well as describing, right? And you want to make sure that you upload your sketches to Blackboard. Okay, you can either upload these as a JPEG file or scan them and create a PDF from them. Okay, but you want to make sure that you include these sketches. Okay, when we're finished with these Are You With Me's, you're going to come back to me. I have a couple of questions for you. Okay, so let's go ahead and look at some of these questions. So here's the first one. A diploid cell has blank the number of chromosomes as a haploid cell. Does a diploid cell have 1/4, half, twice, or 4 times as many chromosomes as a haploid cell? Take a minute to think about that. And hopefully you answered twice, right? So let's go back to humans for our example. In humans, our haploid cells are our gametes, right? Egg and sperm cells. They have 23 chromosomes each. If we look at our somatic cells, they're going to have 46 chromosomes, 2 times the number. So a diploid cell has twice the number of chromosomes as a haploid cell. We could have reversed the question. We could have said a haploid cell has, and you would have said half the number of chromosomes as a diploid cell. Okay, so make sure you know chromosome number. Diploid is 2 times or twice as many chromosomes as a haploid cell. Or we could say a haploid cell has half the number of chromosomes found in a diploid cell. Second question. An organism's traits are determined by the specific combination of inherited cells, genes, proteins, or chromatids. An organism's traits, are they determined by what cells are inherited, genes, proteins, or chromatids? Hopefully you answered blue, genes. It would have been better actually if I put alleles there, right? Forms of the gene, right? So an organism's traits, what color your eyes are, or how tall you are, or whether or not you have a widow's peak, or your blood type. That's going to be determined by the genes that you inherit from your parents. Okay, next question. Seems like my camera keeps moving and it keeps mirroring. So I'm hoping that this is okay. Next question. The first level of DNA organization in a eukaryotic cell is maintained by which molecule? Okay, so that first level, you've got your linear DNA. What happens to that linear DNA? It starts to become packaged or condensed. What is that first level? What is the molecule involved in that first level of packaging? Is that molecule called cohesin, condensin, chromatin, or histone? Okay, so that first level of DNA packaging is going to involve histone proteins. Remember, the DNA gets wrapped around the histone protein, and you end up with those beads on a string, right? Nucleosome linker, nucleosome linker. Okay, that molecule involved in that they're called histone proteins. Okay, last question, and I see a typo. So I'll fix it, okay, while I talk through it. So you got to remember what I'm saying here. So we have a single-stranded a single double-stranded circular DNA molecule describes the genome of which organism below? And I have white button mushroom twice, so let me correct that. So let's say in red, the first answer, let's say that's E. coli, which is a bacteria. Okay, does that describe E. coli? Or does it describe the genome of a giraffe? Or does it describe the genome of the white button mushroom? Or does it describe the genome of the oak tree? Okay, so if you're confused here, let me help you. So again, that first one would be E. coli. That would be a prokaryote. A giraffe would be an animal, which is a eukaryote. A white button mushroom would be a fungus, right? Belongs to the kingdom fungi. And that would be a eukaryotic organism. And lastly, the oak tree, that would be a plant belonging to the kingdom Plantae and the domain Eukarya. Okay, so the giraffe, the white button mushroom, and the oak tree are all examples of eukaryotes. And our first one, E. coli, would be an example of a prokaryote. So which one of those organisms has one single double-stranded circular piece of DNA as its genome? So hopefully you chose the E. coli, the prokaryote, the only prokaryote there. Everybody else, the giraffe, the white button mushroom, and the oak tree, those are all eukaryotic organisms, and they would have several linear double-stranded DNA molecules. Okay, so I think we'll break here, and I'm going to create a separate video for Chapter 10, Part 2.